The marginal zone of the white pulp, which is at the border of the white pulp, is a highly organized region whose function is poorly understood. It contains a unique population of B cells, the marginal zone B cells, which do not recirculate. These appear to be resting mature B cells, yet they have a different set of surface proteins from the major follicular population of B cells. For example, they express lower levels of CD23, a C-type lectin, and high levels of both the MHC class I-like molecule, CD1, and the complement C3 fragment receptor CD2135. Marginal zone B cells may have restricted antigen specificities, biased towards common environmental and even self-antigens, and may be adapted to provide a quick response if such antigens enter the bloodstream. They may not, for example, require T-cell help to become activated. Both functionally and phenotypically marginal zone B cells resemble B1 cells. Recent data suggests they may even be positively selected much as B1 cells are. However, they are distinct both in location and in surface protein expression. For example, marginal zone B cells do not express high levels of CD5. The marginal zone also contains dendritic cells as well as two different types of macrophages, one of which lines the marginal sinus, whereas the other is scattered throughout the marginal zone. B cells and T cells are not the only cells in their respective areas. The B cell zone contains a network of follicular dendritic cells, FDCs, which are mainly concentrated in the area of the follicle most distant from the central arteriole. Follicular dendritic cells have long processes from which they get their name and are in contact with B cells. They differ, however, from the dendritic cells we have encountered previously in that they are not leukocytes and are not derived from bone marrow precursors. In addition, they are not phagocytic and do not express MHC class II proteins. Follicular dendritic cells seem to be specialized to capture antigen in the form of immune complexes, complexes of antigen, antibody, and complement. The immune complexes are not internalized, but remain intact on the follicular dendritic cell surface, where the antigen can be recognized by B cells. Follicular dendritic cells are also important in the development of B cell follicles. Beyond this, their functions are not entirely clear. Have a great day.